So this started out as recognizing gene sequencing and gene therapy. One of the sort of common themes when you're giving a talk is to know your audience. And after yesterday, I decided that we just needed to do gene therapy. Fortunately, Dr. Longo did a great job with gene sequencing, so you're not really missing anything that I would have said about it. So, just conflicts of interest disclosures. I don't have any personal financial stakes in any treatments under development. I have sponsored research. I've had or have sponsored research agreements with the following companies, which you all heard yesterday, so no surprises there. Quick overview of the talk. We're going to talk about what gene therapy is. Are the CCDSs good candidates for gene therapy? How is gene therapy performed? And what's standing in the way of gene therapy for creatine deficiencies? So what is gene therapy? Gene therapy is basically a way to modify genes either by restoring gene expression, eliminating gene function. You can think of eliminating gene function in cancers. So if you have a growth factor that's out of whack, you can introduce a gene to hopefully eliminate that. And then fixing gene mutations. So one of the first gene therapy in human trials was conducted last year by Brian Kaspar's lab at, as we say in Cincinnati, an Ohio State University, not the Ohio State University. So this is single-dose gene replacement therapy for spinal muscular atrophy in these patients with a mutation in the SMN gene. They introduced an AAV, an adeno-associated viral vector, with a full length of the SM1 gene, and the results were that a single dose of AAV resulted in longer survival, superior achievement of motor milestones, and better motor function than historical cohorts. This was a small study. SMA1 is a spinal muscle atrophy. Patients live about 15 months. So this was a good candidate caused by a single mutation or a single gene mutation. So it's a good candidate for gene therapy. The fact that they can extend life gives it a further impetus when it comes to getting it into the clinic a little faster. So how is gene therapy performed? As I said, we use a viral vector with the healthy gene, infuse it intravenously, and that healthy gene then is then expressed in the cells that take up the virus. So the first thing that, when you're talking to people and they ask what you do, yeah, I'm working on a viral vector. And they say that we're introducing to these mice viruses. You're going to make the mouse sick? Well, hopefully not. So a normal virus that makes you ill, basically the cell takes in the virus, it replicates itself, and then either lyses the cell or moves out, making more copies of itself. The viral vectors that we use for gene therapy, we remove a certain part of the virus's genome that is required for it to replicate. The way the virus gets made is we actually put it into cells that express that part of the virus's genome for it, and it doesn't incorporate into the vector in itself. So what happens here is when we deliver this virus, no new viruses. So whatever cell takes up the virus expresses the virus, and that's it. It doesn't replicate and spread to other cells. So hopefully no pathogenic response. There is a slight there will be an immune response to it. So in the first few days, there might be a fever, chills, flu-like symptoms types of things. But for the most part, after that, there should be no general side effects. So are creatine deficiency syndromes a good candidate for gene therapy? The short answer is yes. They're monoallelic diseases. It means they're caused by one gene. They have a clear phenotype. They can be identified using gene sequencing, so we know what gene is affected. We do know through Dr. Solomon's lab that restoring at least creatine transporter function in fibroblasts from CTD patients increases creatine uptake. When we restore creatine transporter function in mice, the first thing you notice, so in the purple here are mice treated with a virus-containing human SLC6A8. The blue line are just control mice. All are on creatine supplemented water. The first thing you notice is an increase in weight gain. So, so when we look at the brains of these animals, the first thing that we notice, so this is a human gene. When we do expression studies in the lab, now we target, instead of targeting the area that we knocked out and getting false positives, or false positives, we've actually started to target the front end of the gene. So it turns out that our animals do express the front end of the gene just a little bit. It's degraded and not made into a protein, so that's why you see this little bit here. But in our knockout mice treated with the creatine transporter virus, you see an increase in creatine transporter expression. When we supplement after 14 weeks, we do see an increase in creatine content in the brain. You'll notice that it doesn't achieve the levels of their wild-type brethren, but they're significantly higher than the knockout. So we'll talk about why this is in just a second, some of the limitations of this. So the other one is that we do see some small cognitive improvement in the mice, but a lot of it was, there wasn't a whole lot. So here's the novel object that I talked about yesterday. There's no difference between the two of those animals. We didn't include any wild-type controls in this, just because this was a smaller pilot study, and we didn't want to lose some of the direct comparison. Our hypothesis was the transporter going to make a difference or not? So here we have the cued version of the Morris maze. This is a slightly different than what I showed yesterday. This is the Morris maze, but we flag the top of the platform so the animal can see it. So they should be able to swim directly to that without too much of a problem. It measures motivation, measures swim speed, things like that. Animals treated with a restored creatine transporter do a little bit better. Could be because they're gaining weight, they're getting stronger. And then when we look at the performance in the Morris water maze, we don't see a statistically significant improvement in either one of these measures. When we look at the probe trial, the animals treated with the virus do get a little bit better, but in the reversal phase, that doesn't hold up either. So we would say that this is a small improvement in cognitive function. So what this shows is that it's feasible, but there's much more work left to go. And we'll get into some of the slides are a little out of order. We'll get into some of the reasons why this could be better in just a minute. So if the first is to replace, the second would be to repair. So the most famous, we'll say, repair right now is CRISPR-Cas9. I think most of us who've watched the news for more than a minute have heard of CRISPR-Cas9. It has been touted by the media as the next great thing. We're going to be able to cure all diseases. We're going to have designer babies. We're going to have everything that we want. We're going to be able to live to 1,000,000 because of CRISPR. Well, that's not exactly true. But let's talk about what is CRISPR. CRISPR is a system in which an enzyme, Cas9, causes a break in the DNA. We use a template from a healthy donor, so this pink right here, to then repair the DNA at that break with a healthy template. So following this cleavage and repair, you have the insertion of your healthy gene. This can also be used to introduce modifications. So if you want to knock a gene out in a mouse, you can use this system to do it. So here's the one thing about this is that, as Dr. Longo said, every cell in your body has the same DNA, so we'd have to do this in almost every cell in your body. So how could it be used to treat CCDS in the future? In theory, it could be packaged into vectors directed to repair the specific mutations. So this is that personalized medicine type of stuff. The caveat here is it must express that guide RNA. So when you look at that yellow strand, it's got to express that. And it has to express the Cas9 enzyme, because we don't have either one of those in our genomes. All right, so limitations. Well, the first one, and the most painful one as far as scientists go, because they, just as an anecdote, Cincinnati Children's hired a CRISPR-Cas9 guy to make knockout mice for us. They promised us that we were going to have cheap, fast knockout mice. So before it would take about, to make a knockout mouse just takes about 4 to 5 months to get founders, if you're lucky, and cost roughly about 15 to 20 grand. They're going to simplify it. We're going to have mice in 2 weeks, or 3 weeks because you had to breed them. We were going to be dirt cheap. It was going to be perfect. We were going to knock out rats. We were going to have everything. It's not as good as it was once advertised. It's shortened the time. It's made it a little cheaper. But what ends up happening is it is very effective, but it'll often put what you want on different copies of the gene. So then you have to go in and do it again. The other limitation, obviously, it needs to be directed to every cell. The final and most important limitation when this comes to translating to humans and where the media hype doesn't really help is that this needs to be turned off after you're done using it. You don't want to be constantly expressing Cas9 and breaking your DNA. So, again, a little out of order. But so all of this that we've seen so far sounds great, so we've got to know what's standing in the way of gene therapy. Well, the first big thing, and as Arjun said, and as we discussed a little bit, yes, there's still a lot that we don't know. The first question is it permanent? Is this something that is going to be expressed for the entire lifetime? Can it be inherited? This is a significant question as well. If these can be passed on to offspring, then there could be some functional consequences of that need to be investigated. And the third part is, will it interfere with the function of other genes? So when you're dealing with an AAV vector, and let me go back here just real quick. Okay, whoops. This healthy gene doesn't insert where the normal gene was. It inserts into a space of those 3 billion base pairs that we talked about before. One of the problems that could come with that is that it could insert into a gene that you need, and it could disrupt that gene's function. Oops, too far. So that's one of the things that we have to concern ourselves with. The second thing standing in the way as far as, especially for creatine transporter deficiency, is the virus doesn't really get into the brain well. And I'll show you a slide about that in just a second. For the SMA1 patients, that's great. That wasn't a big of a deal because the problem with SMA is that it's at the neuromuscular junction. So it can cross into the blood-brain barrier a little bit, but its main target was actually outside. So it does get into motor neurons, it takes in, it's doing a great job. But as far as a neurologic disease, a behavioral disorder, it's not quite there yet, and we're not going to get the expression that we would want. And this is so in there that the cognitive delays are generally caused by large, deep-seated regions of the brain. So you need to get expression in areas like the hippocampus, the prefrontal cortex. You have to have wide levels of expression throughout the brain. And AAV9 is not yet effective in restoring, so this is the vector that they used. When you see this AAV, that's adeno-associated virus, 9 is the serotype, so different flavors of the virus will be more effective in different cell types. So 9 works well in neurons. 5 does pretty well in most cells. There's a variety of different ones. And then we still need to know, and then the third barrier is we need to control when and where the genes are expressed. And this is Dr. Longo's slide at the end actually really helped with this. We don't know what the consequence of expressing the transporter or AGAT or GAMT in every cell would be. And for the most part, when you see these viral vectors being used in animals, we use a promoter that expresses this gene in every cell, expresses it at the highest level that we possibly can. We turn the volume up to 10. We don't want to, that's something that in humans we don't want to do. We want to understand when and where this gene needs to be expressed. So we would ideally want to use that promoter region that the gene requires. So that basically the last point is the delivery of new gene is beneficial, so the replacement is great, but it still would be better to mimic the real-life expression of the gene. So just to sort of highlight this, this is a recent paper. It was published in Nature Biotechnology by a group at Caltech. They used these different viral vectors. They hooked them to a protein that just expresses, or they hooked it to a gene that expresses a green protein, green fluorescent protein. So you can shine a fluorescent light on it, and any cell that expresses the protein will turn green. So here's AAV9 right here. So when you look at the whole brain from the top down, you can faintly see it. When you look at a sagittal section, again, there's that faint expression there, and this is the spinal cord down here, and you can see expression there as well. They developed a new vector, a new series of vectors called AAV-PHP.B and .EB. The PHB was named after their postdoc advisor who recently passed away. And you can see that these two vectors in these animals increase expression compared with the AAV9 significantly. So this is, we thought, man, this is great. This is a new advance. We're going to be able to do some significant things with this, right? So what works in the mouse, remember as I said yesterday, what works in the mouse doesn't always work in the human. So IV administration of AAV-PHP caspid fails to upregulate transduction efficiency in the marmoset brain. Marmoset is a non-human primate. Worse than that, the PHP actually made the animal sick. So it caused hepatotoxicity. So that's a big bummer, right? The thing that we can take out of that, though, is that since it works in the mouse, specifically in the mouse that we have the creatine transporter, or the mouse strain that we have the creatine transporter in, we can use this vector to learn more about this disorder. So it gives us important information that can be used for other therapeutics. So when when one of our pharma partners decides that they've reached a certain stage that they want to do a large-scale testing, we can say, based on what we've seen, you need to start testing it this day, or start treating it this day. Based on what we see, treat this day. This is maybe the target concentrations that we need to hit to see if we can find, to get cognitive restoration. So with that, I hope that out of order didn't mess with too much, but I'd like to thank you guys for your attention. I intentionally kept this short because hopefully it'll generate some discussion. And I'd like to thank you for your attention. Thank the organizers. You guys have done a fabulous job these past 2 days. This has been spectacular. And sources of funding and collaborators. So.
We're all doing gene therapy now.
One question. What in the very first data you presented, where you used a gene replacement for CTD, can you tell us a little bit about what you replaced there? Was it the entire gene in an AAV? Did you? I mean, how'd you get the efficacy from that first set?
Right. So AAV is somewhat limited in how much you can put in. So we can't put. So as Dr. Longo showed, your genes are made up of introns and exons and are very long. So what we actually did with that is we replicated the messenger RNA that's produced, made it back into DNA that took the introns out, and expressed that. So we used basically the 2 kilobase, what makes the protein, and used that to drive expression. And then we titered it with some in vitro stuff that I didn't show to see what we could get as far as the optimal amount of viral genomes needed to replace expression. So,
when you talk about the gene replacement and turning the gene off eventually, so what exactly do you do to shut that gene down from what its intended purpose was?
Yeah, that's beyond a $64 question. So I mean, that's actually one of the main challenges, is how do you get that? So if you're introducing something that's going to repair DNA, if you're going to express a protein that you don't necessarily want expressed all the time, how do you fix that? That's a major stumbling point, and the answer is we don't know. I mean, it's, you know, at least with my molecular biology knowledge, we don't know yet. And that's why that CRISPR has not been the revolution so far that's been advertised, because there's still so much more work that needs to be done before you'd even get to that point where you could have serious bioethical arguments to begin with.